certainly good morning and afternoon and evening to everyone joining us here today. We're live here in Madison, Wisconsin at DNASTAR headquarters for today's webinar on our most recent additions to our Lasergene structural biology suite that in our hopes and our customers' hopes will better support the requirements to support your research that are focused on antibody development, engineering, and protein-protein interactions. My name is Tom Lynch. I'm Vice President here at DNASTAR. Before we get into today's presentation of the new applications, I'd like to take just a couple of minutes to provide a brief overview of DNASTAR and our protein-focused software applications that are helping accelerate the efforts of the molecular biologist. Here at DNASTAR, we're most interested in offering an integrated solution for sequence and structural analysis that covers a broad spectrum of biochemical research and workflows. And for today's talk, I want to start with a phrase that's often heard throughout protein and enzyme research. It's something that was enunciated by the late Dr. Ephraim Racker, who said, "Don't waste clean thoughts on dirty enzymes." Of course, this was later brought to life by Dr. Arthur Kornberg in the 10 Commandments of Enzymology in his 2000 article in the Journal of Bacteriology. So our development team for proteomics applications knows this phrase quite well, as we are committed to developing protein modeling solutions that offer researchers really the best starting point for that work. As you can imagine, with over 30 years experience in the software industry, technologies for sequence analysis have rapidly advanced across all disciplines. The molecular biologist is really being asked to go beyond the gene and into the genome and then beyond that to protein structure and function. Therefore, our vision is to innovate and create sequence analysis applications that are connected to one another and provide seamless access to data, thus for allowing efficient analysis to help answer complex questions. It's not just about creating software for life scientists. It's about creating the most powerful software while keeping accuracy and speed at the forefront of our design. As you may know, we had a major release of our software just a few weeks ago. We're very proud of this release, as with improvements made within our existing applications and with the addition of 3 new programs, we believe that Lasergene version 14 really provides all the pieces to the puzzle for helping advance biochemical research across many disciplines. Before I get into the introduction of our NOVA programs, I wanted to highlight our Lasergene suite with our first group of applications that can be found in our molecular biology suite. In this suite, researchers can access tools for in silico cloning, primer design, Sanger assembly and analysis, multiple sequence analysis alignments not only between genes but also genomes, and then annotations and batch annotations, which are certainly important to improve efficiency of your analysis. In our second group, the Genomics suite, researchers can access powerful tools for next-generation sequence assemblies. We've seen in the last 15 years or so the advent of many technologies enabling researchers to sequence not only very large genomes, but also do deep sequencing along exons, gene products. In the next generation assembly area, we have the absolute most accurate assembler and variant caller. We also provide analysis tools for combining workflows like ChIP-seq and RNA-seq. We also offer researchers the ability to detect variants and have access to variant databases to allow for efficient analysis of your NGS data. And in our third and final group of applications, we offer tools for proteomics research in our structural biology suite. And in this suite, we're offering protein sequence analysis tools and visualization to do structural alignments, take a look at solvent molecular services, protease cleavage sites within proteins, and certainly domain annotation. As far as what our next steps were beyond just the viewing and analysis of protein structures, we started working on a project for structure prediction. And today I'll be talking about our support for protein docking and specific antibody workflows. But as you can see here, we started with the Protean 3D application that allows researchers to explore macromolecular structure, function, and motion. This is really an integrated and graphically rich software allowing researchers to visualize and analyze not only the structure of proteins, but also sequence data simultaneously. And really in Protean 3D, the analysis methods allow you to apply predictions not only on secondary structure, but also for other biophysical properties. And this was really just the start of our recent efforts to better support proteomics workflows. And to continue that effort in our focus on structural biology, it was brought to our attention a tool, an algorithm called iTaser, that was part of a group at the University of Michigan in the Dr. Yang Zhang lab that was routinely dominating an automated structure prediction competition known as CASP. It's a biennial convention. Its acronym is the Critical Assessment of Structure Prediction. And as you can see highlighted here over the last 5 competitions, Dr. Zhang and his lab have experienced a great deal of success. And DNASTAR was very fortunate to have built a strong partnership with Dr. Zhang and the University of Michigan over the past few years in bringing their iTaser algorithm into our structural biology suite as our NovaFold application. So before we get into docking and antibody focus, I just want to kind of highlight a brief background on NovaFold. It utilizes a unique hybrid approach to structure prediction that uses a combination of protein threading and ab initio techniques. Here at DNASTAR, we're always working to better our products and offer the advantage of expert technical support. And since bringing NovaFold to market, we've improved the ability to handle large data sets through the implementation of not only locally available resources, but also really the unlimited ability of cloud computing power. So therefore, in our NovaFold application, we can allow researchers access through a command line program on Linux for the advanced user, but also through the Protean 3D interface, where predictions can be sent to the Amazon cloud and therefore not taking up any of your local resources, and really making a more universally ready product. With NovaFold and with all of our Nova products, we wanted to make sure as we're doing the development and the design that we delivered results in an easy-to-use format. For instance, in the NovaFold report, we deliver the top 5 model predictions with scores. All the information on the threaders that were used for the structural prediction, as well as the protein structures used in the process, are available. Also, we make it very easy to align your structural models with any of the structures used in the prediction, as well as any related proteins. And finally, provide the option, because most researchers want to go beyond the structure in order to predict any ligand binding sites or the potential function of your protein. So kind of as we look to the future of structure prediction and how we're developing software here at DNASTAR, we're working towards developing really a complete program that allows researchers to access many workflows. In our development process over the last several months, we've identified a couple areas in which we need to move forward on. The first was providing a tool that specifically helped antibody-focused research, and the second was the docking of 2 proteins. So I'm going to do, in the second half of this presentation, I'm going to focus on antibody-focused research. And so the development team kind of expanding on the NovaFold product, we have introduced recently in the last few weeks our NovaFold Antibody program for the modeling of antibodies and antibody fragments. Now, unlike NovaFold, this is actually a program that utilizes homology modeling, in which a DNASTAR provided library of known IG structures can be used as a template. We also provide the user access to this library so that proprietary files can be used as well. Now, the thing that's unique about this application is that for loop modeling, we actually perform ab initio modeling for the H3 loop and select from a library the optimal canonical loop structure for the remaining 5 CDR loops based on really a sequence profile. So once the model is generated, you have an all-atom energy minimized structure. We also take the next step to provide easy, really access to information analysis by automatically annotating the CDR loops, which are really the most interrogated site on an antibody, really. So we're directing researchers straight to those most important sites. So when we look at a representative cartoon of an antibody structure, really NovaFold is going to help with the modeling of variable fragments, heavy chains, and fabs really are the most common structure that are predicted by researchers already using NovaFold Antibody. And here we have an example, keeping in line with the presentation of our Nova products data is the NovaFold Antibody Report. And as you can see, this is a fab fragment. It's a prediction with the heavy chain in red, light chain in yellow. And on the right, you can see in the features table, if you look closely, the 3 CDR loops on each chain are identified and annotated. And in NovaFold Antibody, this is a very fast-moving algorithm. The predictions just take a few minutes to run, and of course, in keeping with our mission of continuous improvement, our development team is actually already working on further refining our loop modeling algorithm that's been implemented so far. So now that we have, the ability to generate structural models of antigens with our NovaFold product, and we have the ability to create models of IGs using NovaFold Antibody, really the next question is now what? A lot of times we hear from researchers who are working on either the development of diagnostics or development of antibody-based therapeutics, or want to create novel antibodies and finding and identifying the best epitopes on antigens is, okay, once I have all that information, how then can I interrogate the structure of the protein-protein complex? And keeping in line with our mission to produce, the most accurate and powerful software applications, we are fortunate yet again to build, a strong relationship with Dr. Paul Bates at the Francis Crick Institute in the UK to commercialize what they've developed in-house called the SwarmDock algorithm. We have since brought that code into our house, into DNASTAR, and have rebranded the SwarmDock algorithm as NovaDock, keeping in line the Nova brand of our structural biology suite. As with iTaser in the CASP competition, SwarmDock has been a top-performing tool at the Capri competitions, which is similar to CASP for protein-protein docking. And really one of the main reasons why SwarmDock has been so successful is that flexible docking generally requires an ensemble of rigid structure conformations. Whereas with SwarmDock, now NovaDock, protein flexibility is explored through normal modes of analysis to induce conformational changes upon binding in the simulation. So really this is a kind of a new way of thinking, new way of doing high-resolution docking, and we're very proud of this new application. Really looking at kind of the output of the NovaDock prediction is that this algorithm presents superior models, plain and simple, of the protein-protein complexes, in which then our NovaDock report presents the top 10 poses of the ligand and receptor. And these poses are represented graphically, and alternative conformations, based on the ranked models are also available to study. And in order to keep the analysis efficient, we highlight these poses both graphically and in a tabular format, where we present the side chains from each, the receptor and the ligand, that are thought or predicted to interact with one another. Here is a snapshot of the NovaDock report. As you can see, kind of on the left, it's a little bit small view of the report itself, but as you can see, on the left, you have a couple of things expanded. One is we have a model overview, and then we have model 1 expanded, and then below you can see model 2, 3, 4, and 5. But in the model overview, this is an interactive report where the receptor is presented in its ribbon structure in green, and then the ligand is represented as a bubble. This bubble is the center of mass of the ligand, and as you can see, we have scattered bubbles of green color, orange, yellow, red, and really what's that saying, and they're of different sizes, and what's that saying is that it's like a stop and go light. Green is good, yellow is caution, red is stop, pretty much not the best. But we're presenting the top 10, and in this case, we can see a clear representative green bubble. It's very large based on predicted energy score, and that is what we consider in this program the best pose. If you open up model 1, you get both the ligand and the receptor highlighted in ribbon format. Also, in tabular format, as you can see, you have the predicted intermolecular contacts of both the ligand and receptor highlighted by residue. On the right here, I just kind of have a blow-up of the model overview for closer view. So really, kind of immediately after prediction, we're directly guiding researchers to the key focal points. And in this case, we present a real-world example of a NovaDock prediction. This is a beta-lactamase binding to an inhibitory protein. So as you can see, we have beta-lactamase highlighted in green. We have the inhibitory protein in yellow. And as you can see at the interface, automatically NovaDock will present the side chains that are thought to interact with one another at the interface, highlighted, so it kind of takes a lot of the guesswork of this. And what we've done here is this was actually a known co-crystal structure at really high resolution, 1.7 angstroms. When we did a structural alignment of the NovaDock prediction of the two proteins in a complex against the co-crystal structure, we found that we had an RMSD value of less than 1.3 angstroms, which is telling us that not only did NovaDock perform quite well, but it performed at a really high level of accuracy and resolution. Keeping in line with really a majority of a lot of the research today looking at antibody-antigen complexes. This is another real-world example of a factor H protein bound to bactericidal antibody. And in this case, we set the CDR loops of the antibody as the proposed receptor contacts. Prediction is run, and then in the report, as you can see, side chains are highlighted. Again, the coloring receptor, which is in this case is the antibody, is highlighted in green, and the factor H protein highlighted in yellow. So moving forward, we're always working towards not only improving our NOVA applications and the way in which results are presented, but also how researchers can access the programs. As of today, our NovaFold application is available both locally on Linux and as well as on the cloud through the DNASTAR cloud. and NovaDock and NovaFold Antibody are currently locally run applications, but our team is hard at work in getting these applications on the cloud as well. At this point, I would be happy to answer any questions that anyone has. Kind of open it up.
Hi, Tom. Thanks a lot for that great presentation. We do have some questions from our attendees that they sent to us via chat. The first question is, how long do predictions take for NovaDock or NovaFold?
Sure. Yeah, for most predictions in NovaDock, it's typically on the order of 12 to 15 hours. For NovaFold, prediction of an average size protein will take on the order of 15 to 20 hours. And this is using relatively modest Linux workstations with modest amounts of memory.
Thank you. The next question we have is, can I link NovaFold or NovaDock to my custom workflow? Yeah. So if you want to set up multiple predictions or to link predictions between these applications, they can certainly be scripted for efficiency. Thanks for that. And the last question that we have for today is, can I run my predictions across multiple grid computers? So for that, it's not available yet, but certainly, as I kind of mentioned before, job scheduling can be used to schedule predictions on either individual computers or individual nodes. We're certainly working on being able to distribute these calculations across many instances. Yeah. All right. Well, that takes care of all of the questions for today. And I'd like to thank everybody who attended for joining us. And also thank you to our Vice President, Dr. Tom Lynch, for presenting the webinar. We hope that you found the information helpful. And if you missed any of the webinar, we'll be sending out a link so that you can view it again or view the part that you missed if you'd like to. And if you have any additional questions or have ideas for future webinars, please email those to us at webinars@dnastar.com. Thanks again for joining us, and we hope to see you again next time. Yeah, and to further add to that, Sharon, thank you very much, and thank you all who attended today. I hope that you found this to be helpful. If you have interest in maybe a demo of the program or accessing the trial version of the software, you can feel free to contact me directly at lynch@dnastar.com. Be happy to help. You know, in a lot of cases, researchers have sent us proteins that they wanted to test docking or test structure prediction. We've done that and presented it via demo webinar, but also we have a lot of researchers accessing the trial software in-house to give it a go. So be happy to offer access to the programs either way. I look forward to hearing from you all, and have an excellent afternoon.